All right, welcome back to Science 30. We're still in Unit A on Chapter 2, and today's lesson is Lesson 2.4, and we are looking at mutations and genetic diseases in this lesson. So by the end of this lesson, you should be able to do some of these outcomes here, except this one is actually for the next lesson, so ignore that one for now. First off, we're going to take a look at what are mutations in DNA and how they can affect proteins in humans and result in diseases, genetic diseases. We'll look at some of those genetic diseases. And then we will also take a look at how bacteria can develop resistance to antibiotics and some of these other terms here. There are your textbook readings, so you might want to check those out. If you're running into any issues, take a look at those pages for review. All right, so what are mutations? Mutations are small changes to the DNA sequence that occur naturally, so these definitely naturally occur. But they also occur in more frequency when your body or cells are exposed to high energy radiation or chemicals. And we refer to these substances that cause mutations as mutagens. So these can occur when there's an error in DNA replication. So typically that's when you're going to get mutations, is during replication. And this happens about for every time you're replicating your DNA and you're bringing in those new base pairs. Every 1 billion base pair, you're going to get a mutation. And you'll get more of them if there are mutagens in the picture as well. Now, really important, if these mutations happen in a gamete cell, so when meiosis is happening to form gamete cells like eggs and sperm, that's a problem because then that mutation can be passed on to the next generation. If a mutation just occurs in your cell during mitosis just to create more cells and grow and fix, then it's not as big a deal. But when it happens in meiosis and forming an egg or a sperm cell, then it is a bigger deal because that mutation can be passed on to the offspring. So there's some examples of what mutagens might look like, and some of these are going to come up again in our course as well when we do some looking at radiation, EMR spectrum. So X-rays, gamma rays, UV radiation, all those are going to come up again. Nuclear radiation, cigarette smoke, environmental poisons, there's a bunch of listed ones there as well, and food preservatives, nitrates. Right, so we're going to look at 3 different types of mutations. The first one is actually a mutation that has to do with your chromosomes and the chromosomes and how they don't properly separate during meiosis. So when they don't properly separate, it's called non-disjunction. So normally in meiosis at that first step here, this would be normal meiosis. So you have a pair of chromosomes and one copy from each pair is put into the 2 first new cells when they split. So in non-disjunction, they don't properly separate, and you'll end up with 1 cell with all the chromosomes and 1 cell with no chromosomes, so that the resulting egg or sperm cells have no chromosomes at all. And then the other egg and sperm cell have extra chromosomes. Fantastic. This non-disjunction can also occur at the second step of meiosis, where they separate once again. My image here, and you can see you again have, here's a gamete with 1 chromosome, that's the usual number, but now we have 1 with 2 and 1 with none. So this is where you can get that chromosomal mutations, and when these egg or sperm cells combine, they may not form a viable offspring, or they might cause something like Down syndrome. Remember, we saw there's an extra chromosome 21. Well, that's how you would get an extra chromosome, through non-disjunction. So basically, they're just not separating properly. The chromosomes aren't separating properly during meiosis. All right, so other types of mutations that we're going to look at, probably into more detail right now. The first one is called a point mutation. It's just involving one base pair being substituted with another during DNA replication. So if you have a sequence of nucleotides, A, C, G, C, C, A, just one of those nucleotides is being subbed for another one. So instead of an A, you have a C, and then the rest of that DNA chain is the same. So usually these mutations just result in a slight alteration in the proteins, and they're usually still functional. Although we will look at one where it's not functional anymore. So there's an example of a point mutation up top here. This is your normal DNA sequence here, and the reading frame, it shows, remember, every 3 nucleotides is basically equal or codes for an amino acid, an AA amino acid. And so you would get the normal protein with this top regular DNA code. Here's an example of a point mutation when that G, so the C has been turned into a G. You can see that. And you're still having the same reading code or the same frame, right? These first 3 nucleotides are going to code for the same amino acid. This next 3 codes, they're going to code for a totally different amino acid though. Right, so there's a change in the protein. The next 3 nucleotides, though, are coding for the same like a new amino acid. Actually, the same one as the first one, but there was no change, and there's no change further on down the line. So the protein is all same except for this one amino acid. Now, sometimes that amino acid's not going to matter. It's not going to be a big deal, and other times it'll be a huge deal, and it can cause a big problem. So that's a point mutation. So one nucleotide in the entire sequence has been switched. Then you can also have frame shift mutations. So frame shift happens during replication where they might have added an extra base into the chain or deleted one of those bases. All right. So in the example here, you can compare the original sequence up top with this new sequence, and it all goes along fine and dandy until here. There was a T, you can see that, and the T is missing. So this is called a point deletion. So at that point, that base is gone. So you know how you read normally, you would take the first 3, that's your code for your first amino acid, and then the next 3 bases would give you your next amino acid. Well, you don't have that T there, so now you have to use that C into your code for the amino acid. So then your next amino acid would be these 3. So you shift the frame of the reading of the 3 code has shifted now, and so now from this point forward, all your amino acids are going to be different, or there could be a termination code in there as well, so your protein is shut down. So this disrupts the whole reading frame when a point deletion occurs. Or the same thing happens if you put in an extra base. It shifts all the way that the code is read from that point forward. So that's why it's called a frame shift mutation. These types of mutations usually result in radically altered proteins that are no longer functional. So these shifts, these cause big problems. All right, so types of mutations, we looked at these already, and there is some examples here if you want to look at this again, and there's also a video there to watch if you're still a little confused with these 3 types of mutations. All right, so next we're going to look at bacteria and antibiotic resistance, and these mutations in bacterial cells can actually cause bacteria to become resistant. And then this resistance is passed on, this ability to resist antibiotics is passed on to other bacteria through these two ways below. So some of these mutations are just totally random, but they help and benefit this bacteria to become a superbug so that it can resist almost all forms of antibiotics. So bacteria have two ways that they can transfer that resistance, that DNA that contains this awesome mutation for them. First way is called through transformation. And so what bacteria can do is actually just take pieces of DNA that just happen to be in the environment into their cells and bring it right into this plasmid, this circular DNA that they have in their, it's separate from their chromosomal DNA that they pass on. And then there's conjunction. So some bacteria, they have these plasmids, and they can actually pass the whole plasmid from one bacteria to the other. So they form kind of a channel that they can pass their plasmid DNA and just pass it on that way. So they don't actually have to reproduce to pass on DNA in this case. All right, so this is called conjunction and sharing of genetic information, and yeah. All right, so let's go back to some of these genetic diseases that can be caused in humans by these mutations. The first one, we've been talking a lot about hemophilia, that's the ability to the inability to clot blood. And we've also mentioned way back in that first chapter about sickle cell anemia, and then Huntington's disease. So we're going to take a look at these, a couple of these, and how they occur. So if we start with sickle cell anemia here, you'll see a sickle cell, so hemoglobin is normally made up of 2 alpha chains and 2 beta chains. Okay, and these are made through our DNA. So these are just proteins. Okay, so these are proteins that are coded by our DNA. And here's the gene for it. Okay, and there's our first base, second base, third base. So there's a code for the first amino acid, code for the second amino acid. But you can see what happens down here. There has been a substitution. So normally the base is a T, but it's been substituted for an A. And that is a point mutation that has actually caused a whole disease disorder in humans. Okay, sickle cell anemia. So because of this one point in mutation, this hemoglobin, the beta chain hemoglobin, is deformed slightly, and this deformation, because of this one base change substitution, actually causes this whole disease and causes hemoglobin to be all deformed into that sickle cell shape. Remember we talked about this? And then it kind of gets clot. It can cause clotting in the blood. And it doesn't carry the oxygen very well, and people can get very sick. Okay, so that's an example of a mutation causing a genetic disorder in humans. So now, are all these mutations bad? No. Remember survival of the fittest when we were doing Science 20 or back in Science 10? You don't get variation in a population if you don't have mutations too, right? So you need that mutation to get the variation. So you get that mutation where all of a sudden a couple of birds, a bird born with long beak has this amazing advantage over birds with short beaks, right? That could have just been caused by a mutation. Same with the fur color. Slight variations or mutations might create a variation that is going to give that organism a better chance at surviving and reproducing than other organisms. So mutations can be very good and have led to survival of organisms too. All right, so again, there's a great review video. This Amoeba Sisters is kind of funny. It's a good, that goes through all the different types of mutations. All right, genetic diseases. So, and how are they inherited? Let's just take a look at some of this. We did talk a little bit about this in our sex-like traits, but we're going to get into some talk about pedigrees and how we can look at how these genetic diseases are passed on in a population. So if you are an individual who possesses the genetic disease but does not develop symptoms, those, remember, are called carriers. And remember, carriers are heterozygous, right? They're going to have the normal, let's say H, and also the recessive, a copy of that gene or allele. So carriers are going to pass on that. So in this diagram, there's just a picture of what the carrier is going to look like, and this individual here is going to be affected and have the disorder. Let's get into some pedigrees. Good. Okay, so pedigrees can be used to show how these traits are passed on in a family. So they don't have to be used for genetic disorders. They can be used for any kind of trait that you want to look at. But let's just take a look at what some of the symbols mean in these pedigree charts. You actually have in your data booklet a table that lists like the meaning of a lot of these symbols. Basically, if you have a circle, first off, a circle is a woman, so a female, and a square is a male. If there's a horizontal line between them, that means they're married. If there are lines coming down from their marriage line to another male or female, that would be a daughter or some offspring. So a daughter or a son. Okay, so they're offspring. Now you can see their daughter here, who's a circle, has married a man. Right, so that's showing that she's married him, and he's not attached to them, so he's not their actual biological son, right? He's just married into the family. And then their line down here says that they have offspring, and there is their first son. So this is also important. So when you have children, your first children start on the right-hand side. The second child was a daughter. And then the third child is a son. Alrighty. Now, if any of these squares or circles are shaded in solid, that means they are affected. They're an affected individual. So whatever you're looking at in that pedigree chart, if you're looking at a disease, if you're looking at just a trait like widow's peak, if you have the widow's peak, you have the affection, you're going to be shaded in that color. Colored in. And if you're not shaded in, you're just open like that, light colored, it means you are not affected by the disease. So you could be a carrier. We don't indicate necessarily carriers in pedigree charts. All we look at is, are they affected or not affected? And then from that information, we can determine if there are carriers. All right, so this example just has, I'm just gonna move this up here, has some questions to go with this pedigree, which are very similar to questions you're going to get with other pedigrees in the course. So first off, so this is the Lee family, and we're looking at dimples versus no dimples. So we're not talking about like a disorder here or anything. If they have no dimples, then we're going to consider that's dimples is affected, so shading. So if you are shaded black, then you have dimples in this pedigree. So they want us to label the generations of each member of the generations. Okay, this has been done in blue. So this is generation 1, 2, and 3. And then each member of the generations, you're going to number from the left to the right by age. So there we go. So how many children did I1 and I2 have? These numbers are so we can refer to them. I1 and then I2. How many kids did they have? There are 4. 1, 2, 3, 4. Remember, these ones are all just married into the family. Okay. So they have 4. What was their sex? Okay. Female, circle, square male, and circle. So 3 daughters and a son. And which is the oldest child? The oldest child, remember we mentioned, is always on the right-hand side. So 2, 1 is the oldest child. Assuming that having dimples is the recessive trait. So recessive means the only way you're going to have dimples is if you have 2 recessive traits. So using D's in this case, so DD. So having dimples is DD. And no dimples is the dominant trait. Write the genotypes for all the individuals on the pedigree. Now this has been done, but how has this been done? How do we know that this number 4 is heterozygous, like big D and little d, or a carrier for dimples, and not just 2 big Ds? So we have to kind of work backwards. If we look at those individuals that have the shading, they have dimples. So the only way you're going to have dimples, the only genotype you can have is little d, little d. So all these little d's, double little d's, homozygous recessive genotypes can be drawn for any of the offspring or individuals that have a solid shading, including dad up there. All right. So then how do we know that mom is not big D, big D? 2 dominant alleles. Well, if she was big D, so let's put her gametes over here in a Punnett square, and dad's gametes are little d, what would all the offspring look like in this case? Pause and see if you can think about what they're all going to look like. Pause the video and then come on back. All right, so big D, little d, all the offspring are going to be the same. They're all going to be heterozygous. But they would all look as though they had no dimples. And is that the case? Well, no. If you look at the first daughter, she's got dimples. She's got 2 little d's as her genotype. The only way she's going to have 2 little d's, she's going to get one of those from dad, and the other one has to come from mom. So mom is a carrier, but mom doesn't have dimples, so she can't be 2 little d's. So she has to have a big D and a little d. I hope that makes sense. She's got to be a carrier. Plus, you can see this daughter here has a big D and a little d. Little d would have come from dad, but big D had to come from mom. So another way we know that she had 1 big D. So that's how we kind of go backwards in the pedigree to figure out the genotypes of the different offspring. How do we know this offspring here could not have had 2 dominant alleles? Well, one of their alleles had to come from dad. Dad has dimples. He has to be only having the recessive allele, so he can only pass on a little d. But this individual does not have dimples, so they have to have the dominant no dimple gene as well. So they have to be heterozygous. All right. So what is the chance that the next child of individuals 2-1 and 2-2, so let's see, 2-1 and 2-2 will have dimples? So let's do a Punnett square. This Punnett square is really messed up over here. So let's do a Punnett square for these 2 individuals. So we've got DD. I'll put them along the one side. The 2 gametes that D and the female 2-1 could have. So this is female 2-1. And then the male or the dad is heterozygous, so D and D. And now let's check out what the possible offspring they could have are. So one offspring could have, so 25% of the offspring could have a heterozygous genotype. Here we're going to have a homozygous recessive genotype. Here we're going to have heterozygous genotype. And here we're going to have homozygous recessive genotype. So it looks like 50% of the offspring are going to be heterozygous, which would be what kind of phenotype? No dimples, right? And then 50% are homozygous recessive, and they will have dimples as their phenotype. One more question here. What is the chance that the next child of 2-4 and 2-5 will be a boy with dimples? Well, if we take a look, and we're just going to do a new Punnett square for this. They both are dimple homozygous recessive parents, so they can only pass on the homozygous recessive gene. So all the offspring are going to have dimples, aren't they? But look at the question. Boy. What is the chance of they're going to have a boy with dimples? Well, remember what the chance of having a boy is right off the bat? 50%, right? 50% chance of boy, or 50% chance you're going to have a girl. Every time, doesn't matter what. Now, all the offspring are going to have dimples because all they have is recessive genes. So you have a 50% chance of having a boy, then you also have a 50% chance having a boy with dimples because all their boys are going to have dimples. Okay, so we've looked at a little bit about this in the past. Let's pull it all together and look at a few more examples here. So remember, autosomal traits, these are not sex-linked traits. So these are, doesn't matter if you're a boy, girl, you can get these no matter what. They're not carried on the X or Y axis, these X or Y chromosomes, these two here. If it's a dominant trait, autosomal dominant, that means both homozygous dominant and heterozygous people with those genotypes will both be affected. Because it's a dominant trait like that, it's not going to skip generations. It's going to show up in every generation if there is a if we're looking at this trait, and the affected only have to have one affected parent. Okay. Now, if you're talking about an autosomal recessive, you are only going to be affected with this if you are homozygous recessive. Okay, and so it can skip generations. It may hide for a few generations, as we saw, and show up again, and because there's going to have carriers in the population. So this kind of thing, you're going to have carriers. And these are both carriers here. 2 carriers can give you an affected child. So you'll skip a lot of generations because of carriers. Okay, so 2 unaffected people may have an affected child, and there's your example of 2 carriers. Okay, and then we have, remember, sex-linked traits that it can be dominant and sex-linked traits that are recessive. So a sex-linked trait that's dominant. All right, here is an example of a genotype of an affected person when you have a dominant sex-linked trait. You can have, again, there's 2 genotypes. You can have a homozygous, so they're both the same, homozygous dominant, and heterozygous, big A, little a. Both of these are going to be affected individuals. Same as above here. Okay. Males are mostly affected in these cases. and all daughters of affected fathers will have it. So because affected fathers are going to have the dominant allele that causes the trait, that's the only thing they can pass on to their daughters is that X chromosome with the dominant trait on it. And why are males mostly affected? Well, males are only getting one X chromosome from their mom. And if it has the trait on it, then they will be affected. Okay. Whereas a daughter who gets the trait from their mom might get the other X that's not affected from their dad, so they could be a carrier more often. All right, and then there's sex-linked recessive traits. So if you get the recessive, so as a male, you only need the one recessive allele to have the trait. As a female, you need both recessive to have the trait. Okay, and in this case, males are mostly affected, and girls must have an affected father, and all sons of affected mothers will have it. Let's look at some more detailed examples. So here's an autosomal recessive example. So remember, autosomal, anything not on the X or Y chromosome. We're going to look at the Punnett square of what it could look like for this. So this example is for cystic fibrosis in humans, and it can skip generations because it is recessive. Okay. Let's take a look here. So we have a female who has cystic fibrosis and a man over here who has cystic fibrosis. So the parents 1 and 2 here, I1 and I2 generation, the father does not have cystic fibrosis, but the mother does. They have offspring. None of their offspring actually have cystic fibrosis, but they're all going to be carriers. Okay, so there's no, let's just use a little r here. Because it's recessive, this mother would be passing on that recessive gene to each one of her offsprings in that case. So they're all carriers now. Over on this other side, I3 and I4. So this man here, he's got cystic fibrosis. Since it's recessive, the only way you're getting that is if you have 2 recessive alleles. So he also has passed on a recessive allele to each offspring. If these two find each other and mate, and they each are carriers of cystic fibrosis, then their offspring, you can see, most of them are not affected by cystic fibrosis, but in this case, one is. So they have there could be a 25% chance. But let's take a look here. 3-2 is a girl that suffers from cystic fibrosis. Yeah. Other than her paternal grandparents and maternal grandparents, all the other relatives are phenotype normal. So you would not necessarily know cystic fibrosis ran in that family unless you had that pedigree and that history and that chart to look it up. So that's what the pedigree might look like. You might see a whole generation where there is no trait showing up, and then it shows up again later on. So this is what a typical autosomal recessive pedigree might look like. Autosomal dominant example is you're going to see that trait show up in every generation because it is a dominant trait. So it's going to keep showing up during that pedigree. A sex-linked recessive example. So when it's recessive, women who are heterozygous are carriers for the recessive allele. So if you're like that, you're a carrier in this case, right? Except we're talking sex link now. So that's how we would write it for autosomal, but now we're talking sex link. So if you're a female, you might look like that if you're heterozygous. So you are now a carrier. So women can be carriers for these recessive examples. So they can pass along that recessive allele to their son, and that's how their son would express that gene. So you're going to see sons more frequently with this, with the traits, and more frequently when you're talking about sex-linked recessive examples. Okay. So you got some females that are marked as carriers in this pedigree, but only the males actually have the diseases. So this is a very common, very popular sex-linked recessive example during the hemophilia was passed on throughout the European royalty during the 19th and 20th centuries. So Queen Victoria of England, through two of her 5 daughters, passed this mutation on to other royal houses across the continent. And you can start to see hemophiliac males. You can start to see them in this pedigree here, and the ages that they died. It's quite sad. Yeah, so there's a lot of hemophilia in that that royalty line that got passed on. But again, the point is that you can see that it's affecting the males. There's carriers that are females throughout, but no females that actually even have the hemophilia. And then there are very, very rarely, but there are a few examples of sex-linked dominant traits, and this is where you only need, so if you're a female and this is a sex-linked dominant trait, you'd no longer be a carrier like you would be with a recessive. Now all you need is that one dominant allele and you've got the disease. So then if this is the case, you're going to see this disease quite frequently in all men and women in your pedigree. All right, this video is a good review of all the pedigree material that we've been covering. I suggest you watch, take a look at it, and then these are the resources that were used to create this.